phylogenetic systematics. If you could explain to us what about the phylogenetic systematics, what about genetics, genetic similarities demonstrates universal common ancestry. Go ahead.
Phylogenetics demonstrates that the bigger the difference between you and some other animal, the farther you were separated. So the longer you were separated from your common ancestor. So, so compare a human genome to a mouse genome, they would have been the last mammal split off, because mammals were originally mouse-like, and then they split off and stuff. So the farther apart the genetics or genetic difference, then the farther your ancestors splitted.
Now, and that's a good point. Now, are you familiar with the fact that phylogenetic systematics, as you're talking about here, it's mostly based on the concept of outdated junk DNA? So since functions are only beginning to be found, say during the last decade, meaning we actually understand less than 5% of the DNA language, the trajectory, and I will also say that a lot of the data is preliminary, but the trajectory is most important. It strongly suggests that the vast majority of DNA will be functional. Now, why that does not bode well for phylogenetic systematics is because this takes away the primary assumption of evolutionary phylogenetics, which is actually based on mutations for one and pseudogenes, right? Genetic markers. But if the vast majority of DNA is functional, as we're seeing now, Godless Galaxy, it now becomes a common design argument. And what's nice and revealing is that creationists have always predicted that the vast majority of DNA is functional. So this functionality is certainly a problem and inconsistency for phylogenetic systematics. What do you think?
So you're saying that because we do not completely understand each function of DNA, that's like one of the reasons why we can't use phylogenetics?
Well, because a lot of these, phylogenetic trees, they're based on genetic markers that are assumed to be, mutation related, or let's use pseudogenes, for example, plagiarized mistakes or what they would call them, right? And if they really are plagiarized mistakes, that would certainly be strong evidence for universal common ancestry. But in regards to the ENCODE project, they found out that at least 80% of the non-coding DNA was actually transcribed into RNA, right? So that actually suggests function, but it's also been found that these non-coding regions, and most of our genome is actually non-protein coding, I'm sure you're aware of that, but it's now known that these regions of genomes correlate with biological complexity even better than the protein coding regions. So that actually suggests that these non-coding regions, Godless Galaxy, they carry significant information, say for regulatory activity. But a big thing as well as ENCODE, they didn't look at the portions of the genome involved primarily and exclusively in development. So that type of DNA would only have function then. So that means the trajectory of discovery favors genome-wide functionality very strongly. Probably, there's certainly a lot more function in the genome than evolutionists formally assumed. But the point is if these genetic markers are actually functional DNA differences, they can't be used as plagiarized mistakes to build these phylogenetic trees. That's the point. You know what I mean? That's the point. So now these trees just remain in the realm of hypotheses. Right now, that's why the battle, rages on. Did ENCODE really detect function, or is it just spurious transcription? But if it really is function, once again, genetics points to common design. Like, do you understand what I just said there?
Sort of. So you're saying because these markers, they were claimed to be mistakes or whatever, but that's a way to disprove evolution?
Well, because a prediction based on, pre-existing heterozygosity would be that the non-coding regions are functional. Okay, but evolution says that there will be much non-functionality. So they use a lot of those, say, pseudogenes, which are actually in those areas, as genetic markers to build their trees. But like I said, in regards to ENCODE, the ENCODE project, these results have suggested that at least 80% of the human genome is functional. But as I said, is the results are preliminary. Right? I've talked to scientists on this. I wouldn't want to straw man the ENCODE and what some of the evolutionary scientists are saying. They'll say that a lot of it could possibly be spurious transcription. But one thing is clear is that most non-coding DNA is at least not idle. It's doing something. Now, why would the cell be wasting so much energy transcribing if it's not really doing any useful activity? So I think that that's kind of a rescue device on behalf of the evolutionists. But I think the whole point is, if these pseudogenes, if these genetic markers that are being used to build the phylogenetic trees are functional, then that means these phylogenetic trees are just based on a false assumption that molecules to man evolution is true. But now we know it's not true. So they really are just lines on paper. And lines on paper don't prove a thing, right?